Welcome to the PubMed Daily Review for March 07. Today we scanned 2,113 new open access papers. Here are the top 3 selected for detailed analysis. Up next, our first paper out of 3, contrast-free identification of glioma blood-brain barrier status via generative diffusion AI and non-contrast MRI by Kaiyi Zheng and Liming Zhong from Biomedical Engineering, Southern Medical University, Guangzhou, China. Let's break down its key contributions. Assessing the blood-brain barrier, or BBB, is crucial for understanding and treating gliomas. Traditionally, this requires contrast agents like gadolinium, which can have side effects. What if we could get this vital information from standard non-contrast MRI scans, making the process safer and more accessible? To tackle this, researchers developed a novel AI model called CBSI. It uses generative diffusion networks to synthesize contrast-enhanced MRI images from standard non-contrast scans. This allows the model to predict the BBB status without any contrast agent, aiming to improve accuracy and safety. The results were impressive. On an external test set, CBSI achieved an AUC of 81.31% for BBB status identification. This represents a significant improvement of over 11% compared to using only non-contrast MRI. Importantly, its performance was comparable to models using actual contrast-enhanced MRI, demonstrating its potential. This research offers a promising way to assess glioma BBB status without contrast agents, potentially reducing patient risks and healthcare costs. By providing this critical information from standard scans, CBSI could improve diagnostic accuracy, aid in treatment planning, and ultimately benefit patient care. Up next, our second paper out of 3, Ubiquitin E3 ligase KLHL6 brings exhausted T cells back into action by Zhuiyao Chen and Benedict M. Kessler from Medicines Discovery, Nuffield Department of Medicine, Target Discovery Institute, University of Oxford, Roosevelt Drive, Oxford, OX3, 7FZ UK. Let's break down its key contributions. In cancer and chronic infections, T cells that are constantly stimulated can become exhausted. This exhaustion prevents them from effectively fighting the disease, and it's a major hurdle for many immunotherapies. While we have some treatments, they don't work for everyone, so scientists are searching for new ways to reawaken these tired T cells and boost the body's immune response against tumors. To understand what drives T cell exhaustion, researchers used advanced techniques like CRISPR screens and single-cell RNA sequencing. They compared activated T cells with exhausted ones to pinpoint key molecular players. Their work focused on ubiquitin E3 ligases, a class of proteins involved in protein degradation. This approach led them to identify KLHL6 as a crucial factor that seems to keep T cells from becoming terminally exhausted. The study found that when KLHL6 was lost, T cells became exhausted, showing increased levels of proteins like TOX and PGAM5. However, when KLHL6 expression was restored, it significantly improved the T cell's ability to fight tumors. Even inhibiting a pathway called PI3K-AKT, which normally suppresses KLHL6, helped bring back KLHL6 levels and reactivate exhausted T cells, demonstrating a clear link between KLHL6 and T cell function. This research opens up an exciting new path for cancer treatment by identifying KLHL6 as a critical regulator of T cell exhaustion. The findings suggest that we could develop drugs to boost KLHL6 activity, potentially re-energizing the immune system to fight cancer more effectively. This could lead to new therapies that benefit a wider range of patients and might even have applications beyond cancer, impacting other immune-related diseases. Up next, our third paper out of 3, calcium release channel deficiency syndrome in patients diagnosed with idiopathic ventricular fibrillation and decedents classified as sudden unexplained death in the young by Lucilla Giamarino and Michael J. Ackerman. Let's break down its key contributions. Sudden cardiac arrest and death in young individuals, especially those with no clear cause, are devastating. A recently identified condition, calcium release channel deficiency syndrome, CRCDS, linked to specific gene variants, might be an underlying cause. This syndrome can lead to dangerous heart rhythms without showing obvious signs on standard ECGs, making it difficult to diagnose. To investigate this, researchers looked at two groups: 169 patients diagnosed with idiopathic ventricular fibrillation and 279 young individuals who died suddenly without an explained cause. They performed genetic testing on the RYR2 gene, searching for very rare variants that could be linked to CRCDS. This was a retrospective study, analyzing existing clinical and genetic data. The results showed that 3% of patients with idiopathic ventricular fibrillation carried RYR2 variants potentially causing CRCDS. This rate increased to 9% for those whose sudden cardiac arrest was linked to exertion. In the sudden unexplained death group, 11% had RYR2 variants, and importantly, 7% of exertion-related deaths in this group were linked to loss-of-function RYR2 variants. This study suggests that CRCDS is an important, often overlooked, cause of sudden cardiac events, especially in young people and during physical activity. Identifying these RYR2 variants through genetic testing is key for accurate diagnosis. This knowledge can lead to better patient care, allow for screening of family members, and help prevent future tragedies.